Chapter 10 in histology deals with the anatomy and the histology of the male reproductive system. And the first slide illustrates a conceptual diagram of the functional features of this system. Anatomically, the business end of the male reproductive system consists of seminiferous tubules, which basically make up the bulk of the testis. Those seminiferous tubules are the site of male gametogenesis, whose stages we'll document in a few moments. In essence, their secretory products, sperm cells, leave the seminiferous tubules in the testicle, and they pass through a series of ducts that allows them to enter the head of the epididymis that you see here. From there, sperm and seminal fluid components, particularly sperm, pass through the length of the epididymis down to the tail of it, which marks the origin or the beginning of the ductus or the vas deferens. The vas deferens then ascends from the scrotum, passes through the inguinal canal in the lower anterior abdominal wall, passes posterolaterally to the male bladder, and then posterior and inferior to the bladder, the ductus deferens joins with a duct of a seminal vesicle on a corresponding side, and the two structures together form an ejaculatory duct that then empties into the lumen of the prostatic urethra. So by the time sperm has entered the prostatic urethra, it has been mixed with seminal fluid secretions provided by both the seminal vesicle and the prostate. And then sperm and seminal fluid are released through the prostatic urethra, through the membranous urethra, through the penile urethra, and into the outside world. Anatomically, our first task is to visualize the structure of the seminiferous tubule. And you can see in this schematic diagram that literally these form a tubular array of a stratified epithelium that contains not only the cells undergoing various stages of gametogenesis, but they are also lined from base to luminal surface by a support cell known as a Sertoli cell. So in the area of detail, this is shown in a schematic view on the subsequent slide. First off, we want to make the point about what cells are situated closest to the basement membrane and what is the progression of gametogenesis and the change in cell type as we move from the basement membrane to the luminal surface of the seminiferous tubule. Note that lying in direct contact with the basement membrane are the primordial germ cells prior to puberty in males, and that at puberty they proliferate by mitosis and form type A and then finally type B spermatogonia. So these cells basically are always in a position where they are situated adjacent to the basement membrane. Then, once a type B spermatogonium is committed to meiosis, and meiosis 1 particularly, it passes through a blood-testis barrier, where basically that blood-testis barrier, as illustrated here, is formed by tight junctional contacts between adjacent Sertoli cells. And once the spermatogonia enter meiosis 1 and form a primary spermatozoa, they pass through that blood barrier and are now protected from autoimmune destruction by that barrier. Primary spermatocytes, once they complete meiosis 1, will form secondary spermatocytes. Secondary spermatocytes, once they complete meiosis 2, will form spermatids. And you can see as we go through the successive stages of meiosis in males, the resultant germ cell moves closer to the luminal surface of the seminiferous tubule. Spermatids are haploid entities that then undergo spermiogenesis to form a mature sperm, whose tails obviously are projecting out into the lumen of the seminiferous tubules and whose heads are still embedded in the apical cytoplasm of the Sertoli cells. Note also, and we'll see this histologically, that the Leydig cells that produce testosterone basically are situated in the interstitial connective tissue that's found between adjacent seminiferous tubules. Now, the details. As we said, basically, cells that undergo spermatogenesis are going to be arranged in anywhere between 4 to 8 layers that occupy the space between the basement membrane and the luminal surface of the seminiferous tubule, and only the Sertoli cells span the entire width from the basement membrane to that luminal surface. The stem cells, as we say, are the primordial germ cells that lie in a quiescent fashion adjacent to the basement membrane until puberty. At puberty, they commence mitosis producing more primordial germ cells that also differentiate into type A and then into type B spermatogonia. Once they reach the type B stage, then they are committed to enter meiosis by forming primary spermatocytes that will pass through the blood-testis barrier and subsequently be protected by it. Now, in terms of the stages and the details about what's going on, primary spermatocytes basically replicate their DNA, so they transform a complement of 2N DNA to 4N, and they're still diploid. But note that the prophase stage that includes the first stage of meiosis involving primary spermatocytes is very long. It's a process that lasts anywhere between 10 days and 2 weeks. And at the end of that period, basically, a primary spermatocyte will form a pair of 2 secondary spermatocytes, basically dividing their chromosome number in half, rendering them haploid, and dividing their complement of DNA and obviously subdividing into 2 secondary spermatocytes that are both 2N. The next issue is the secondary spermatocytes rapidly undergo meiosis II in a matter of minutes. And as a result, secondary spermatocytes are very rarely seen in a typical histologic section that includes the gametogenic cells. So once secondary spermatocytes complete meiosis II, they form haploid spermatids that are basically maintain a chromosome number of 23, but since they don't replicate their DNA, the DNA is reduced from a complement of 2N to 1N. And note that the progeny of a single maturing spermatogonium remains connected to the other spermatogonia by cytoplasmic bridges throughout their differentiation process into mature sperm cells. Our next slide shows a histologic section through a portion of a seminiferous tubule stained with H&E. And it illustrates, again, the histology, easily recognizable, of the fundamental cell types that it contains. The arrowhead marks the location of the basement membrane that, among other things, consists of myoepithelial cells. Immediately adjacent to the basement membrane are the spermatogonia, indicated by letter B. Then, once a spermatogonium is committed to meiosis I, it forms a primary spermatocyte that passes through the blood-testis barrier and is now situated at letter C. And what you see at letter C, in addition to the one that's marked by that letter, are a number of primary spermatocytes that are in various parts of meiosis I, mainly in the prophase stage, which is the longest segment of it. Then, the remaining cell types with smaller nuclei that move toward the luminal surface are all spermatids, smaller, more condensed nucleus, and then projecting out into the lumen of the seminiferous tubules are the tails of sperm cells with their heads still embedded into the apical cytoplasm of Sertoli cells. Note that, finally, there is a pale staining elongate nucleus with a prominent nucleolus at the tip of the arrow, and that's the location of the nucleus of a Sertoli cell, which are the support cells, and as we said, extend from the basement membrane to the entire luminal surface of the seminiferous tubule. The next diagram shows a mature sperm cell. And anatomically, a mature sperm consists of a head and a tail. The tail has 2 components, a midpiece, which is the energy source for sperm motility because it contains the mitochondria. The principal piece contains the array of microtubules in the form of a trusilium, whose motility is maintained by a dynein and ATPase motor molecule. The head of the sperm basically consists of 2 components. It has the nucleus and the acrosome. The nucleus contains the haploid genetic material that are the result of meiosis I and meiosis II. The acrosomal cap is derived from the Golgi apparatus of the spermatid, and it contains the hydrolytic enzymes that are associated with the acrosomal reaction that'll permit sperm penetration of the zona pellucida at fertilization. Next, let's look at the interstitial area between the seminiferous tubules. And here, as we indicated schematically earlier, we find the testosterone-producing Leydig cells at the tip of the arrow that are interspersed among blood vessels and additional connective tissue elements, including fibroblasts, one of which is illustrated at the tip of the arrowhead.